Okay, hi, hello everyone, and my name is Xiangyu from Griffith University. I'm a little bit different from the PhD student. I'm graduated. So, yeah, I'm a little bit ashamed that I'm still consider myself like a PhD student here today, maybe last time. But yeah, and I was supervised by Professor Chen and Lucas, and also the Mara. And I think Lucas just mentioned that my PhD is more about to looking at the matrix or generating the matrix and the indicators of soil resilience. And what Lucas just mentioned is all about the incubation experiment. And in your PhD, I always worry that if I test the theory in lab, it's okay, but it cannot really work in the field. That's going to be a problem. So we take a little step further and to try this resilience concept and simplify it, just looking at the soil responses to the compaction in the field. In a larger field, it's about 80 hectares. So back to my today's presentation, I'm going to talk about the sugarcane soil response to the compaction and the removal of compaction. So to the experiment design, this study is only working on the compactions, and also there are different field measurements to removal the compaction and looking at how the soil microbes respond to the compaction and its removal. There are 4 treatments. There is a control is control, compacted soil, and also another treatment is about meal mud shallow furrow addiction treatment, which means we put the meal mud about 10 centimeter depth. And the third treatment, just called the deep trench treatment, we cut the soil about 25 to 40 centimeter depth. And also the fourth experiment is about the deep meal mud application. After we cut the soil deep, and we put the meal mud at around 25 centimeter depth. Okay, and these 4 treatments were divided into 6 plots, and also soil core were randomly sampled with the 5 centimeter diameter auger, with divided into 4 layers. 0 to 10, 10 to 20, 20 to 40, and 40 to 60. Okay, so yeah, like I have mentioned, we have 4 treatments, and we are looking at how those treatments affect the physical, chemical, and biological properties of the soil and the related nutrient cycling. And then this soil properties will further affect the soil health and also the productivity. It's really important to link our theory to the productivities, because it's not really that persuasive talking to the farmers like, oh, you please believe my theory, but I cannot guarantee your productivity. That's going to be a problem. So it's really important for me to try to link my theory into the productivity. And these are the sugarcane soil, so the productivity here is related to the sugarcane yield and sugar yield. Okay, so in this experiment, we have 3 hypotheses. One of them is meal mud application will increase the soil nutrient pool, and also different application method will lead to different distribution pattern of the nutrient. And also the removal of compaction will increase microbial activity, which will further improve the soil health and increase the yield. Okay, let's look at some results. Yeah, we can see here after we surface apply the meal mud, there is significantly higher content of total phosphorus and also coil phosphorus, which is as our expected. And also when we deeply apply the meal mud, they are increased the total phosphorus and also the coil P according to the And also, besides those nutrients, we're also looking at some microbial properties. And we can say that with the deep application of MILMAT, we found that entire soil profile has increased microbial carbon content. And also, like I've mentioned, that surface applied MILMAT will increase the microbial biomass carbon and nitrogen at the surface area. And also, it's really interesting, we found that deep application of the MILMAT, they changed the microbial carbon and nitrogen ratio a lot. This could be a good indicator to indicate that the soil microbial community is shifting from, like, for example, from the bacteria-based into fungi-based. So this could be a really good indicator. And also, we finally linked our activity, our experiment theory to the yield of the sugarcane. And also, we found really interesting that in the deep trench treatments, without any MILMAT application, they also showed an increased yield. This was really interesting. And also, after we gathered all of the results, we did a PCA analysis. We found at the surface area 0 to 10 depth, we found the surface applied the MILMAT treatment are separate from other treatments. And at the little bit deeper, we still found the surface applied MILMAT treatment are different from others, but others start to separate from each other. And also, okay, on my way, okay. So in the 20 to 40 centimeter depth, we found that the deep application treatment are separate from other treatment. And also, most interestingly, at the most deepest layer, the deep trench and deep trench MILMAT application treatment are separate from others. So there are some conclusions I can make based on our experiment. So the first one is application of MILMAT improved the soil condition by stimulating the nutrient cycling, and also enhanced the soil health and crop yield. And also deep trench application of MILMAT stratified the nutrient supply to soil profile with the microbial community in all soil layers. And also, we found that deep trench field management improved soil physical conditions, which enhanced the nutrient cycling and also further improved the soil health and the sugarcane growth. And at the end, I would like to thank to Soil CRC and also to the SRA and the Burdekin Productivity Service, Rob Miller, and for their support to this experiment and my entire PhD. Thank you very much.